back can hear me.
Yeah.
Are we starting right on the spot, or should we hang?
Let's start right on the spot. Let's do it. So we are thrilled to be able to introduce Dr. Tom Oliver, who is the team lead for the NOAA Coral Reef and Climate Change group. Tom's been around the block. He did his undergrad at Cornell, where we were housemates, and then went on and did his PhD at Stanford. Did a stint doing NGO work in Madagascar before returning back to be faculty at UH, and then moving on to his position at NOAA. Did I forget anything there?
That's pretty much it.
Got it all.
That works.
And so Tom's really on the front lines of investigating how climate change impacts our coral reef and all the animals and people that survive off of those coral reefs. He's going to tell us a little bit about his efforts right now.
Okay, great. So to get the full job title, there's like 8 different organizations for whom I officially work for, but at NOAA, in the Pacific Island Fisheries Science Center, there's all these scientists that are studying fisheries. These days, fisheries actually means ecosystems, right? So a big chunk of that group studies coral reef ecosystems. They're based here, but the Coral Reef Ecosystem Program studies coral reefs in all of the US Pacific. So what is in the US Pacific?
Water.
Hawaii is in the US Pacific, that is correct. What else?
Alaska?
Alaska is in the US Pacific, but this is the Coral Reef Ecosystem Program. So we mainly focus on shallow water, scleractinian coral reefs. There's a lot of small islands south in the Pacific that we've lumped together into a new monument called the Pacific Remote Island Areas.
American Samoa.
American Samoa is one. Yeah, so American Samoa and the associated islands. That's Tutuila, the Manua Group, Swains Atoll, and Rose Atoll. The Northern Marianas, we also spend a lot of time in, so Guam and the islands around Guam. And the Papahanaumokuakea as well, right? That's the Northwest Hawaiian that is now a monument as well. So basically my group out at NOAA does a lot of work to monitor the status and trends of that whole region. But that's not what I'm going to talk about today. What I'm going to talk about, so this is the natural history of the Hawaiian Islands, yeah?
Yes.
And, whoop.
Yes, all says yes.
Yes,
Your
good.
questions.
So what I'm going to talk about today isn't not so much focusing on an organismal group, but I'm going to talk about coral reefs as an ecosystem, and one of the tools that we use to study it, and that tool is genetics. So basically, the topic of today is coral reef genetics, and why you might want to use genetics as a tool to study the ecology and the conservation of these systems, right? So the cool thing about genetics, though, is most of what I'm going to say while I spend my life studying coral reefs is relevant to just about any other ecosystem that you might want to think about. So in the natural history of the Hawaiian Islands, if you want to talk about tree snails, these things apply, right? The same kind of questions using the same kind of tool. So, and actually Anthony and I do a lot of parallel work, even though he studies fungus and I study coral reefs. We've actually published a paper together on the funguses on the coral reef, all using these kind of tools. So to start, can anybody tell me what this is?
The graph.
That is a graph. That is important observation. All right, so it is a graph that runs from 1971 on the x-axis all the way to 2011. And what is on the y-axis? Transistor count. What does that have anything to do with it affects our lives in any way? Okay, I'll just let you out of the box. So this is what's called Moore's law, right? And you see the number of transistors packed onto a microchip since the 1970s until a few years ago has increased in this linear fashion. Is that a linear fashion? It looks like a linear fashion, but we are lying to you. Because this y-axis is what's called a log scale. So you can see that's 2,000, that's 10,000. And then the next 2,000 is that big. So that's 100,000, a million, 10 million, 100 million. So that is a very exponential growth. Right? Moore's law basically says, over this whole time period, the power of a computer chip has doubled about every 18 months. Okay? And this is what is responsible for the fact that we now have way more computing power than took us to the moon in everybody's pocket, right? And if you think about all the ways you interact with computers, right, that this kind of change, if you think about the way that your parents and or grandparents interacted with computers in 1971, and the way we do now, it's because of Moore's Law, right? The power changed so fast that it could come and affect our lives. Yeah?
So the laptop I bought in 2014 may only be half as powerful as the ones that are out of work now.
Possible, yes. And these days, power means a lot of different things, but it would, processor speed and transistor count, you are correct. So that's Moore's Law. Anyway, I don't want to talk about Moore's Law. I want to talk about genetics. So this is another graph. And this is the cost of raw megabase of DNA sequence. Notice the tricky y-axis, right? So what this means is that back in 2001, there was a moonshot of a project, which was to sequence the entire human genome, right? To figure out all of the genetics for a human, right? To do all that sequencing. And that takes many megabases of sequence. A megabase is just a unit of sequence. It literally means 1 million DNA bases, right? Like the letters of DNA. But the cost of doing that went down and down and down, right? Pretty quickly. That white line is Moore's Law, the same rate that we saw computers change our lives over 30 years, right? And so in the first few years of this graph, it was tracking that amazing, mind-blowingly fast rate of technological and societal change. And until about 2007, it did even better than Moore's Law. And you can see just from where the scale on the graph goes, that around 2007, there was a major technological shift that led this cost of DNA sequencing to drop dramatically, to the point being that what cost $10,000 back in 2001, now the unit cost is a dime. And it is actually, this is an old graph, so really it's now pennies on the dollar, right? So the speed of that change has already, in the 5 years since this happened, really transformed the way that we approach questions in biology, right? And it has made tools that were previously only accessible to study fruit flies and mice to basically study any organism you find of the diverse array of obscure organisms you'll find in special places like Hawaii, right? So even here, if you find an endemic organism that no other scientist has come near, you can be up and running studying its entire genome in the timescale of months because of this change. Okay, so what that means is it gives us a lot of power. And these are the two platforms that really were responsible for the dip in that curve. We don't have to get into the details of it. But that's my question to you today. Okay, that's cool, that's fast, it's going to change our lives. But why do we care as biologists who are focusing on studying the natural history of the planet?
Better technology to do your jobs.
Yeah, better technology makes our job easier.
And the cost is lower?
The cost is lower, right? So that means there's things that we just, on the budgets that we have, we couldn't do before. But maybe a better question, as opposed to why do we care, is what can we do with genetics in this system? Or in any system? Yeah?
There's like so many species, different species, vegetation species throughout Hawaii. And we have some like that much data to see the relationship. If there's a relationship.
Absolutely can, yeah. So that is a great point. That actually sets me up for my next slide. But before we go to that one, any other ideas? Like what else could we do with these data?
For conservation purposes?
Uh-huh. Like what? There are lots of ways we can use it for conservation purposes. You're totally right. But can you think of specific ways?
As, like as far as genetics or as far as saving money or both?
Well, probably for like what genetics could you do? And you don't have to have the answer. It's okay if you don't know. But what I've just told you is that genetics got a lot cheaper. And if you don't care about it in the first place, you probably don't care that it's cheaper, right? So like the cost of mud is $10. Now it's $1. Do you care? Maybe if you really like mud. But
I'm sure there's some way that you can use genetics, like the genetics of coral reefs, to help conserve them.
There are, and we'll talk about it. Anything else? Yeah.
Are you talking about changing the genetic makeup in order to conserve?
I hadn't been, but we can talk about that. It's a very controversial question, but one that has gotten some, especially given the threats that we're facing in coral reef environments right now. No one is talking about GMO anything, but when you say changing the genetic makeup of our reefs, the world is already doing that for us, and there are people thinking about how we can help prepare them to go through that process of adaptation. And we can't do that without understanding the genetics. So, no to GMOs as far as today, but yes to changing the genetic makeup of reefs. Yes. Any other thoughts? So, the first one is what we were just talking about in the back there. What is this?
Darwin's first phylogenetic tree.
Darwin's first phylogenetic tree, full marks. So this is basically the first phylogenetic tree that was ever written down. It's not a classification scheme. There are many trees written before this point that were classification schemes. What's the difference?
I think it shows the relationships to the common ancestor.
Yes. So the difference is, classification scheme is what Linnaeus, really, the guy we think of when we think classification, Linnaeus, he said, this thing is like this thing, so I'm going to put them in the same box, right? What's different between that and an evolutionary tree is that it tells us actually where these things came from. It talks about the history of the paths that they walked to get to where they are today, right? That is a phylogeny. It's a description of an evolutionary relationship. And with genetics, while that, tree was not genetically informed necessarily, modern phylogenetic trees almost invariably are. And usually the best trees are a combination of morphology, the shape, and what basically organisms look like, and an increasingly sophisticated array of genetic tools, right? And what you're looking at there is sort of the most recent summary of all life, as represented as a phylogenetic tree. But the cool thing about science, and the cool thing about science during a fast-paced technological, effectively, revolution, is that this tree is being updated. And I put this in the talk a couple months ago. It's already wrong, right? That's how quickly these things are changing. So it's an excellent tool for phylogeny. Another thing that makes it an excellent tool is sort of related to conservation, but it's an excellent tool for studying biodiversity generally, right? So if we want to understand where different species are, what different species are, and what are the processes structuring where you find them, that's the study of biodiversity. And especially in coral reefs, studying biodiversity is really hard, right? Coral reefs are arguably the most species-rich environment in the planet, on the planet, right? And much of that diversity, those individual species, are unknown to science, right? So people frequently talk about, like, oh, we discovered a new species, as it being an enormous deal, right? And that is true if you're talking about a mammal, if you're talking about a bird, right? If you're talking about a fish, it's still a pretty big deal. If you're talking about marine invertebrates that live on coral reefs, you're describing the status of about 60 to 70% of the things that you're going to find just by poking around a bit, right? There are undescribed species all over the place, which means it's hard to know if my job is to study the status and trends of US Pacific coral reefs, specifically including biodiversity. So to do my job right, I have to know what species are where, and preferably know why they're there and how that's changing, right? Without genetics, I couldn't do that job well, right? So one of the ways that we do that is we use something called ARMS, or Autonomous Reef Monitoring Structures, right? So one way you could go out and figure out what's on a coral reef. You can swim on it, you can look around, you can count stuff. That works really well for corals and for fish, right? And we do a lot of that. Corals and fish represent literally less than 1% of the species diversity on coral reefs. If you want to study the other 99%, how do you do that? Any ideas?
You take a sample,
Take a sample. What do you mean by take a sample?
you break a piece off and bring it up and study it.
That's exactly right. Yeah, that's what you have. That's one of the ways that people have done this for a long time, and still one of the best ways to study this is to get yourself a hammer and a chisel, and pick a specific section of reef, and chisel it out, hammer it up, put it into buckets, and carry it up to the top, and then spend days and days and days sorting through everything that you find. Right? You can imagine in this era of global threats facing coral reefs, getting permits to bash up large chunks of your reef, even if you wanted to get those permits, is difficult, right? For good and bad reasons. So we have been trying to use another way, right? By using basically what is a fouling device. So it's a thing, it's this little condo made of PVC that we stick out in the water, and we pin it down to a coral reef, and we leave it there for years, right? Until it gets fouled by crustose coral and algae, and basically becomes a very habitable and happy place for organisms that like to live inside a coral reef to live, right? It's not exactly like a coral reef, but it does a good enough job that and it does it in a comparable way all across the areas where we work, right? That we can wait 3 years, cover it up with a mesh-lined milk crate so nothing gets out, strap it down, pull it up, and basically begin to go through all of these little critters that are in there. So this is a photograph of all of the big, greater than 2 millimeter organisms found in a square meter of reef and moray, right? And you can see there's all sorts of things in there, a whole diversity, right? So we can count those, right? We can look at them, we can take pictures of them, we can clip little pieces of tissue off them and run genetics on them. And that lets us figure out not only what they look like, but also really what species they are and how many different species are represented by this diversity, right? So we can count and we can track biodiversity. But, you know how I said 1% of the diversity is found in corals and fish? Most of the diversity is actually found in stuff smaller than 2 millimeters. And so the way we get at that is with this.
Blender.
We literally scrape the plates of that into a blender, blend it into a slurry, take samples of it, and sequence it, right? Using the same tools that we would use to sequence individual species, we can sequence and basically ask the question of what's in that slurry, and we get information that isn't paired with an individual sample. So it's not quite as good, but is almost as good to tell us who's where on the reefs, right? So biodiversity. And right now, we have hundreds of these units out all over the world, and we are running through and processing them and generating collectors curves like this, where we control for sampling effort, the number of those units we pull back in each of these places, and we can see the taxonomic richness of organisms that come out. How many species are there? Okay, so that's one tool. We can use it to actually study the biodiversity we're trying to conserve. Whoop. I just gave you guys a little window into what's going on next. But so there are other ways that we can use it in a conservation context. So you just saw the word, so I might as well put it back up. So what does that mean, and why is it useful?
They're related to parents.
It is related to parents. So imagine you are studying Nene. Right? Nene is an amazing success story at this point, right? That they were reduced in number and they have been recovering, right? One of the real challenges when you're studying a group like that is avoiding inbreeding, right? So there are certain pairs that if you put them together may not be viable, right? Or will potentially reduce the amount of genetic diversity you have in the population, right? And for a population that's been reduced that starkly, maintaining that genetic diversity is really important, right? So one of the ways that we can do this is that even if you've come upon a population, you can figure out who's related to who, right? And in genetics, we call this parentage analysis, right? So if you just walk into a room full of geese, and you're like, all right, who are first cousins in this room, who are siblings in this room, who are not related in this room. You can take genetic samples and just like you're on tabloid TV, figure out whose daddy is who, right? And it's really useful when it comes to conservation work, especially with large body organisms. It's also really helpful when it comes to the more sort of small critter stuff that we do, that usually have much larger population sizes, to discover how they're moving around. If you can figure out where an organism's parents are, you know pretty well where it came from for most organisms that stick around somewhere, like most of their adult life. And figuring out what populations are linked in space is really hard. And parentage is one tool that we can use to do that. Okay, forensics. What do I mean by that? Bless you. Okay, so you're in Japan. In Japan, they sell whale meat, right? You can buy whale. But they're only legally allowed to sell one kind of whale. South Pacific minke whale, right?
Is that the whale that all those whaling Japanese ships go out to hunt whenever
Correct.
they're on the news?
They don't, correct. They wouldn't call it hunting, they would call it scientific take, because they are studying what the whales eat by cutting open their guts. And then just as a byproduct, they're selling the meat on the open market. So they would call it scientific take. But they're only allowed to hunt and kill and sell that species. If you go to a whale market in Japan and you pick up that piece of whale meat, how do you know it's that species?
You don't. It's just genetics.
Genetics, right? It's basically the only way. I mean, there are ways you could maybe say this doesn't look like whale meat, but to know this is a South Pacific minke whale, when you're looking at a tiny percentage of the entire organism, genetics can help you with that. And now it's pretty old science. Back in the 1990s, when this was first done, folks miniaturized their lab into a suitcase, set up their genetics lab in a hotel room in Japan, because Japan wouldn't let them export the whale meat, and asked that question, what species are present? And it turned out every whale species in the world, effectively, was present in the markets, including dolphin and goat, and a number of other things that were supposedly whale, and supposedly South Pacific minke whale. And because of that work, there's been a lot more scrutiny on that kind of work. Right? So in terms of conservation work, forensics can be really useful. The same thing is true for fisheries work, right? You can imagine if you can sell bluefin tuna to an unsuspecting buyer and it's actually albacore or something, a totally different fish, you can get a much higher price for it, right? And, so both for conservation and for just general keeping the honesty of the market going, this kinds of wild-focused forensics is an important role. Yeah.
So has there ever been any prosecutions in Japan for selling other whale species besides the?
That's a really good question, and I honestly don't know the answer whether it ever got to prosecution. A lot of things changed because of that research. I don't know that anybody was individually held liable in a court of law. I should ask that question. I tell that story a fair amount, and I don't. It's a good question. Okay. The last point on this before I go on to a vignette about sort of another way to use genetics is connectivity. So what is connectivity?
The attachment something has to something else?
Yeah, basically. And in this context, we're talking about populations, right? So if we're talking about conservation in the Hawaiian Islands, this is very relevant, right? If you think about how organisms live, this is really true on land, but it's also true in the ocean, that they live as populations, right? And so if we're trying to conserve and manage them and keep them around, right, we have to think about them on a population scale. What does a population mean?
The number.
Different organisms living together in a given area.
Yeah, different organisms living together in a given area.
Population.
How do you define what area is where I'm going?
Same species.
Sure, let's stick with same species. That's generally what we talk about with a population. It's a species in a given area. But if we're trying to figure out what is our given area for a population, how do we do that?
You have the geographic range.
So knowing the geographic range will tell you where the species is found. but it won't necessarily tell you that this population, right, is different from this population over here. Gene flow, like so connectivity and gene flow are sort of, they hold hands, right? They're sort of two different perspectives on the same coin, two different sides of the same coin. But really, but the direct answer to the question is generally people think about it in terms of area because that's what we've done in the past. We've gone to a place and said, nah, this is, you know, given this organism, this is roughly, we'll call it this population. But really what you want to know is who's having sex with who. That's what makes a population, right? So if adults can migrate thousands of miles and have sex with other adults and produce babies, right? That's a population. That's how you link populations together, right? If the only prospects for a given organism are within 100 feet of it, that's going to be its population, right? So how do you know that? How do you know who's having sex with who?
Genetics.
Yeah, the answer is genetics. Yeah, because basically you can track, like tracking gene flow, right, is basically knowing who's had a lot of sex with who, right? That like if this place is genetically similar to this place, and this place is genetically distinct from both of them, you can quantitatively estimate how much migration and gene flow is going on between these two, right? And to what scale this one has been isolated, right? And so that mapping of populations that are connected, or to what degree they have connectivity with each other, tells us at what spatial scale we need to think about managing these populations. So this has been a really important question in Hawaii for a long time, especially when it comes to marine management, right? So a group at HIMB, we call the Tobo Lab, for Tonin and Bowen, who are the PIs of that lab, have over the past decade or so produced hundreds of studies of the genetic similarities and differences among marine organisms in the whole Hawaiian archipelago, right? And by looking at all these different species, they've really been able to ask the question, all right, what is the appropriate scale for us to think about managing these places, right? And it used to be thought that you need to manage at the scale of the archipelago, right? The main Hawaiian Islands, and then sort of secondarily manage at the scale of the Northwest Hawaiian Islands, right? And one of the ideas that led to the Northwest being protected was the idea that they'll actually be exporting young and larvae, and there will be connected gene flow from the Northwest down to the mains, right? That was about a decade ago. This was what people thought. After all of this research, we've identified that there are a huge number, well, not a huge number, there are about 5 consistently repeating barriers to gene flow, right? The Big Island is different genetically from Maui Nui. Maui Nui, different genetically from Oahu. Oahu, different genetically from Kauai. And once you get up into the Northwest, it gets a little fuzzier, but there are probably only 2 real barriers up there. Those are actually quite well connected. But they don't export. If anything, it goes the other way, right? So we can even look at directionality of connections, right? So if you think by protecting all the Northwest Hawaiian Islands, that's going to give you a seed bank that's going to continue to rain down to the mains, probably not so true. And if you think you can just, you know, think about conserving at the scale of the entire main Hawaiian Islands for marine systems, also probably not true. We have to think about it really from an island scale, right? And so this kind of genetic data that led to thinking about how populations are connecting, who's having sex with who, really changes the way that we have to think about how we write our laws and how we think about what's allowed and how we think about managing species that are targeted and all of these things. And it all came back to understanding that fundamental question about what is a population as informed by genetic data. Questions? Okay, so it is now 1 o'clock. How long do we have?
20 minutes?
20 minutes. Okay, we'll probably do it in 20 minutes. I'll give it at least rough strokes. So there's one last thing that I haven't talked about. That's one of the cool things that that drop in price that I talked about at the beginning has really allowed a major change. Is that now we can not only use genetics in non-model species, things like coral. We can not only use it as a way to study who's related to who in terms of the phylogeny. We can study their evolutionary history. We can study how they're related to other individuals in terms of their parentage. We can study what species they are for biodiversity or for forensics. We can study who's connected to whom, right? But we can also now study what their body is doing at any given minute. We can study the physiology of these organisms and understand something just about how they're responding to their environment, right? And so I'm going to tell you one story, or part of one story, that comes from my PhD and immediate postdoctoral work, looking at corals responding to temperature, right? And so this section from a talk titled Overheated Corals, Transcriptomic Response and Coral Thermal Tolerance. Does anybody have any idea what transcriptomic means? Okay, so part of that word is transcript. Does anyone remember what mRNA is?
Messenger.
Messenger RNA. And when messenger RNA is finally finished being polished and capped and everything, what is it called?
Gene template.
It's called a transcript. Yeah. So, because it goes, you transcribe
Transcribe and
DNA
translate.
into RNA, and then you translate RNA into proteins. And proteins are what do the body of work, the work of a cell is largely done in proteins, right? But the mRNA transcripts, and those transcripts that are present at any given time in your body, give you a pretty good idea about what your body is up to, right? It's kind of looking at the to-do list of your body, right? And the cool thing about that is that they are sequenceable. RNA is easily converted to DNA. DNA is easily sequenced. So we'll get there. But first, some basics about corals, right? So I assume these are things that you know. Corals are a mutualistic, what we call holobiont. And I assume, Cindy, we've talked about corals, yeah? Have we gone?
Tuesday.
Tuesday. Oh, Tuesday. Okay, so I'll give you the sneak preview that I'm sure many of you already know. Corals are animals, but they live like plants, mostly, shallow water corals, right? And they live like plants because they have an algal symbiont in them, right? It's a dinoflagellate, it lives in their tissues, and it makes food from sunlight, and it feeds the corals, and the corals grow. And it's a mutualistic relationship, right? Because those plants, effectively those algae, get nutrients, they get protection from grazing, and they get nice, stable light environments, right? Corals are a really great diffuse reflector. And corals get food, and they also help them build skeleton. That is the fundamental relationship on which all coral reefs and all the biodiversity of coral reefs is built. That relationship between scleractinian corals and their algal symbionts. But that relationship breaks down, as we saw historically this year and last year in the Hawaiian Islands. How many of you guys know that there was a bleaching event this year? Yeah. Do you guys know how many we've had before this year?
2.
2. We had one in 1996, it was a pretty minor event. We had one in 2014, it was a pretty major event. And we had one in 2015 that we're still documenting, but it looks to be an even more major event.
What happened in 2015?
A whole confluence of factors that was a combination of the El Nino, the PDO, and a bunch of just a very hot year.
What was the second one? P what?
It's the Pacific Decadal Oscillation.
Oh.
Okay, we can look it up. But just know that this is a problem. This is called coral bleaching. And what it fundamentally is when corals get too hot, the symbiosis breaks down, right? And either the algae up and leave or the corals kick them out, one of the two. And what it means is the corals don't die, but they get very sick, right? And many of them can die. If the heat goes on long enough, it will kill them. And we also know that it's going to get worse, right? The projections, this is actually a reasonably old slide at this point. The projections really are now running along that red line right now. And even if we did everything we could starting to be the best behaved in terms of global climate change today, we still have 20 or 30 years where it's going to continue to increase probably, right? So this is a real problem that Hawaiian corals are going to face, and they're going to have to deal with it. They're going to have to go through this process of adaptation that we talked about earlier, right? How are they going to do that? Corals have 3 major ways that they do this. 2 of them are really sort of what everybody can do, right? Acclimatization or natural selection. Acclimatization is you get used to it, right? You go through some process of physiological change, just like when you get off the plane here and you go someplace where it's really cold. It's really cold for a while, right? And you actually do physiologically acclimate to those conditions over a period of weeks, right? Corals can do that too, to some degree. There's some latitude for getting used to higher temperatures. And estimating that is a professional challenge. Natural selection, of course, is the other one, where corals that are able to deal with that rise in temperature they'll survive, they'll reproduce. Corals that aren't will just die, right? So that is basically what would happen to anybody. But because corals are a fundamentally symbiotic creature, there's a third thing that can happen, right? Is that selection can happen, but instead of selection happening on the whole coral, it can happen just on the symbionts. Because there are different symbionts that can tolerate different levels of heat, right? And so basically corals that have symbionts that can take the heat, those symbionts will survive. And to some degree, corals have the ability to switch to new symbionts. So without necessarily having all of those corals that couldn't take it die, they can potentially be, those adult corals can live and have their symbiont populations be replaced, right? For that to be true, for that to be an option, this statement has to be true, that symbiodinium genotypes affect the colony phenotype. What do I mean by phenotype?
Their physical appearance. Right?
Yeah, it's basically how that thing is, right? Like a phenotype, like if you tolerate heat, you have a heat-resistant phenotype. If you can't, you have a heat-sensitive phenotype, right? It's kind of what you look like morphologically or how you respond behaviorally to something is your phenotype, right? And so if it's true that different symbiodinium genotypes can affect the colony phenotype, then what I just ran through is a possibility. And turns out it is true. So this is an evolutionary tree of all the different symbiodinium, these algae that live in corals. And you can see that there are two major clades that occur in the Pacific, one of which tolerates heat pretty well. And you can see, we've had it repeated over and over again, that in bleaching events, you see this pattern where the same species right next to each other, one of them bleaches, one of them doesn't. Why is that, right? In some cases, we can explain. In some cases, it's because the guy that didn't bleach has a different symbiodinium genotype, right? And that might allow them to better resist that change. So with that as an idea, a bunch of colleagues and I went to Ofu Island in American Samoa. This is the Mandoa group in American Samoa. And the cool thing about Ofu is that it has these back reef pools. So if you look really close along that side, you can see that there are these different pools, right? One of which is very large, one of which is very small. And their different sizes basically mean that when the tide comes in and out, the small pool, as the tide goes out, heats up a lot faster in the summer heat than that large pool does. And so the corals that live in those pools deal with a lot higher temperatures on a daily basis, right? And so basically that small pool is hot and variable, and the larger pool is a lot cooler. This makes a really great place to go, just to visit. It's beautiful, but it's also a wonderful place to work because you have a really cool difference in thermal environment, right? Right next to shore, where you can just walk out and you can get a whole diversity of corals and sample them. And when it comes to this perspective on how that symbiont is affecting the temperature tolerance, right, you can see that this type D symbiont, the red heat protecting one, is really common. As a matter of fact, it dominates this hot and variable pool, where it's only about 50% of the population in the cooler pool, right? So the symbiont seems to be following that pattern. And so if we're thinking about whether, these corals, we can think about that moving from that large to that small pool as being analogous to sort of waiting for 50 years for the temperatures to heat up. And we can think about a great place to run some experiments to see which of these processes is happening and how fast they're happening. So to do that, we set up a lab and basically ran a bunch of experiments right underneath the palm trees next to these coral reefs. And we published some papers on it. We basically ran an experiment where we took corals from both of those pools, and put them in a tank where it's nice and cool, and put them in a tank where we heated things up by about 3.5 degrees, which is a considerable amount more heat than they're used to. And we tracked basically how they did in terms of mortality and some other measures, but just for the quick one, just showing you mortality, right? Basically, there are 3 groups here, right? There are those corals from the large pool that have the heat resistant symbiont, and the corals from the large pool that have the heat sensitive one, and the corals from the small pool, all of which had the heat resistant symbiont, right? And you can see, if you stayed up at the top, you stayed alive, right? And if you came down, you died. The only one, and the blue lines there are the control tank, right? Where we didn't stress them. Everybody was fine. The red lines are the elevated temperature tank. And you can see that this top one is the corals that came from that hot pool that had symbiodinium D, right? And those are the only ones that took this level of stress. And they did it significantly better than the other 2, right? So corals with both symbiodinium D, right, and potentially that had acclimated to that thermally variable habitat survived as well as controls. So I give you all of that setup just to say, but how did they do it, right? What about those corals makes them different? We can observe that, okay, these guys can take the heat, these guys can't. But we want to know more than that. We want to know what the genetics are that let them do that, or if there was some other physiological response that let them do it. How can we dig into it? We can dig into it with this other technique, with what's called RNA-seq. Okay, so I'll explain that in a second. But basically, we did the same repeated experiment. We stressed them out for 3 days with comparing tolerance to sensitives, and we extracted and sequenced their messenger RNA, right? To sort of look at their to-do list. And let me just tell you how that goes, right? As we talked about, you can take your DNA, right? DNA makes RNA, makes protein. And what RNA-seq does is it literally extracts all of the messenger RNA in an individual organism and sequences it and gives you that sequence, right? And then you can compare that sequence to databases. And it's equivalent to reaching your hand in a jar of jelly beans and pulling one out, and then basically binning them out and saying, well, I've got 42 of those, and I've got 10 of those white ones, and and you can basically count all of the different transcripts, right? And lay them out, and basically seeing what's turned on when, and how those groups that are tolerant are different from those groups that are sensitive, right? Can tell you a lot about how that process, both evolutionarily and physiologically, goes on, right? And just think for a second about how many millions of sequences you need to be able to do that, right? So if you think about that arc of cost that we started with, right? When things were expensive and sequencing was hard, this would be impossible, completely impossible. There's no way you could do it, right? You're trying to sequence a representative sample, at least, of all of the transcripts that are in a complex organism's body, and to do it on the budget that they'll give somebody who's studying coral, right? Totally impossible 10 years ago, hard 5 years ago, and relatively straightforward with even small amounts of funding now, right? So even if I don't get to the end of this talk and tell you how it turned out, that's what I want to leave you with. So from there, we then assemble the transcriptome. We basically get all of those sequences and figure out what basically the functional genome of this creature is. And then we analyze genes that are expressed differentially, meaning genes that are turned on in one group and not turned on in the other, right? And then we can follow through a bunch. The following slides are a whole bunch of results that are kind of pitched. And it's basically a lot of detail. I don't necessarily want to plow through it in the 5 minutes that we have left. But to get to the end of this, let me just go through this. That basically, yeah, here we go. There are a bunch of things that these corals do differently, right? From one group to another. One of them is really obvious, that they have a group called heat shock proteins. right? That are proteins that literally, when you start to fall apart because you're too hot, they hold you together, right? Those were more highly expressed in the heat-resistant genes, but then a bunch of other genes, and like that's something that we could have told you before we started the experiment, right? But a bunch of other genes, that are in charge of basically holding tissues together and in charge of controlling cell death, right? Things that actually get labeled as like tumor and cancer genes, but really are mostly focused on when things start to go wrong and one of your cells starts to misbehave, the organism kills it, right? So you can think about basically any cellular stress you get put into, that's a reasonable thing to do. And if you have differences in the systems that control that, it may mean a lot in terms of your ability to deal with stress, right? And so, we can also take a look at this in the Symbiodinium, and there's a bunch of other places we can go, but I'm gonna go ahead and end it here. But just with the observation, right, that if what you care about is the natural history of the Hawaiian Islands, if what you care about is the biology and the ecology and the evolutionary history of these systems, genetics provides an amazing tool to peer in and to ask questions. And even if you're not interested in genetics, if you're interested in fundamental questions about how these systems and how these organisms live, interact, and understanding how they got to where they are today, I would highly recommend that you look into the possibility of genetics as a tool that you may want to use in the future. So I can end it there, and I'll take any questions that you guys have. Yeah?
Are there any kind of documented cases in Kaneohe Bay of oil switching blades for this fast bleaching?
That's an excellent question. And as for switching, the answer is no. So the work on it, primarily, like that I'm answering your question with, is by a guy named Ross Cunning. Oh, you work with Ross. Okay, great. Yeah. So this debate about whether corals can switch different types of Symbiodinium has been a protracted debate. Some corals can, some corals can't. And in some contexts they can, and in some contexts they can't. What we saw in Kaneohe Bay in 2014 was that basically we could explain about 50%, correct me if I'm wrong, you've been working with Ross, but Ross's data suggested you could explain about 50% of those corals that were bleached and unbleached with symbionts. There's another 50% that we have no idea what's going on and why some were better than others. But you could explain it with symbionts. But what didn't happen, that did happen in a lot of other systems, is when they recovered, in other systems, they recovered with the type D, with the resistant one. In Kaneohe Bay, when they recovered with the same old type. And so if this is a problem where they're going to get whacked again and again, they may not get better. Yeah, fair enough.
Based on your findings, do you think that there's more hope for the survival of coral reefs in the face of climate change into the future because they have the ability to evolve and potentially select for these clades that are more heat resistant?
Yeah, I think that is the fundamental question we're trying to address, right? And the question isn't so much, do they have these abilities, but can this capacity to evolve and capacity to adapt keep pace with the pace of change? And the pace of change that we're seeing now is faster than any that we've seen in at least 800,000 years, if not 55 million years. So can they adapt? The answer is yes. Will they adapt? The answer is they will to some degree. Will they adapt enough to maintain coral reefs as we know them? The jury is still out. Most sources say no. Sorry. But I'm one of the optimists. So there's still the potential. And figuring out how they're going to do it and how we can help them do it is largely my job. Yeah?
Are corals able to be bred and grown in captivity?
They are. Enter another strategy for dealing with corals.
So you can breed like the heat-resistant corals and possibly set them up in the wild to replace.
That's exactly what is work that is ongoing at HINB. So Ruth Gates' lab, where the man in the back has been helping them out, recently received what is a big amount of funding in our world to do exactly that. To basically to go out to see corals, find the bleached and unbleached pairs, sample the unbleached ones, bring them back to a lab, do all this kind of work to study why they are the unbleached ones, and at the same time try to propagate and restore reefs with those. So when We're talking over here about other ways to change the genetic makeup of reefs. Yes, not through genetically modified organisms, but using genetics as a tool to better understand the system so that we can help rear and outplant in exactly those lines. So that's ongoing. When I say a big amount of money for our field, it's still a tiny amount of money compared to the scale of the problem, right? So we very much, many eyes are on that lab and that effort, and assuming it can work, as I think it can, to basically to double down on that effort and do it at a larger scale.
All right, let's give Dr. Oliver a big round of applause.
Thanks, guys.